Hello, and welcome to the Attomarker webinar on long COVID. My name is Jonathan Snicker, and I'm joined by Professor Andrew Shaw, the founder and CEO of Attomarker. Just some housekeeping at the start. This session will be recorded, and the recording will be available later for you to share. We have a very brief poll for you to answer at the start of the session, which should be appearing on your screens now. Thank you very much. Now, the vast majority of this session, we're going to be covering the Q&A. Thank you very much for submitting your questions, and thank you, Andrew, for agreeing to respond. Now, we've had a number of questions. I've tried to group them. They come under certain themes. So let me ask you, Andrew, the first question. Where or how can we access monoclonal antibodies to treat viral persistence in long COVID?
So at the moment, hello everyone, by the way, sorry I didn't say hello. At the moment, the antibodies that are available that might be useful are available only in the EU. The drug is sipavibart. It seems to work well for all variants, although we haven't measured it directly yet, but that's the claim from AstraZeneca. So it is available in the EU. The MHRA are reviewing it at the moment, and they were expected to give an opinion on it in the first quarter of the year. So we're waiting on them to say yes. There's another antibody in America I know about called Pemgarda, which is also being considered for a trial. It's still awaiting approval, but the FDA have removed approval for all antibodies that don't work across all variants.
And could you tell us, Andrew, a little bit about how the test works?
Okay, so the Prof's going to do a bit of a lecture here now. Let me share some slides. So I'd like to walk through some of the ideas that were developed in my research group at the university, and that then turned into the test that's available, properly validated and prepared for Attomarker. Making sure we're on the same page, the persistent virus idea comes from the fact that you get an acute infection. The virus goes into your body and it permeates through your lungs and the rest of your body. And while you're unwell, you get the flu-like symptoms, you get the sickness, and the damaged cells into which the virus has entered get repaired by your entire immune response. At the end of that process, you've got to clear it. And then for a period of time, there may be some ongoing symptoms. It takes a while to clear. And then it leads into the long COVID problems. And it's probably because there are viral reservoirs, and the virus is hanging around your body, in your lungs, in your digestive tract, in your heart, your brain, and causing inflammation, maybe causing autoimmunity, maybe causing gut dysfunction. And that leads on to this 250 symptoms of long COVID. Why so many? Well, the front door for the virus is this thing called the ACE2 receptor, and it will enter cells wherever they happen to be, whether in your brain, whether in your lungs, whether in your liver. All these parts in green have ACE2 receptors, and that's the problem. It's difficult to clear from all these regions. And ACE2 is the front door, and this is the bit on the virus that allows it to go through the front door. It's called its spike protein, and it joins into these ACE2 receptors around the body. So what our test does is measure how well you make these things. These are the antibodies, and it measures how well you make antibodies to the spike protein, not just how many, and you do need enough to get all around to all the corners of the body, but also how good they are. What's the quality of antibody? And that's our novel part of the test. That's the bit that makes us different from the rest. So what the test then, the sticky bit is important because here's the virus. The antibody sticks to it. If it doesn't stick for long enough, and long enough is about 2 hours, we've just learned, then all these processes here that have the immune system attack the complex and then clearing it finally don't work. If this drops off, it doesn't get cleared. Not only do I have to get the antibodies all around your body, the virus is in your head, it's in your lungs, I've got to get a large concentration everywhere. When they find the virus, it's got to stick. So what we've done is to measure the antibody concentration against a spectrum of 14 spike proteins from 14 variants, all the way from Wuhan up to KP3 at the moment, so 2 more than I mentioned on this slide. So what you want ideally is you make good antibody concentrations to all variants. And this is a patient who has had not a patient, this is a person who's had a series of vaccines and a couple of infections, but is making good quantity of antibodies to all variants. And this quality is the important number. Around 50% or so means those antibodies stick for a couple of hours. When we look at the long COVID community, they often have poor antibodies. They didn't make enough to start with, either from infection or vaccine. Maybe that's a general problem. Maybe they're always immunocompromised, but they've got gaps in their spectrum, usually around quality. So here's another long COVID patient here. Quite a good response to the antibodies made from the vaccines. These are the early variants triggered by the vaccines, but none of the later variants are making good quality antibodies. And so if you had an Omicron infection that generated your long COVID, you're not going to clear that from the body. Nothing sticks around for long enough to help it clear. This persistent virus therefore accounts for, we've learned recently, about 65% of the long COVID community. We're about to publish this paper. It's being reviewed by the authors at the moment. We'll go into the journals. And I think this is the big jump. The community divides into groups. 65% have this persistent antibody problem, a persistent virus problem. There's 10% of the community have hyperimmunity. They're making huge concentrations of antibodies. They're all really good quality, but it's not clearing the disease. It's somehow, and we've seen this at 200 milligrams per liter, this is almost the maximum rate at which you can make antibodies. Why is that being triggered? This hyperimmune autoimmune response is about 10% of the population. The antibodies don't go away, and they're not clearing up the symptoms. And then I've got about 25% of the population for whom my test doesn't say anything. They may be suffering from other diseases like Lyme disease or resurgent Lyme disease, but for the moment, I can't tell the difference. So what are the treatment options? If you get your spectrum measured and we find the gaps and they align with your clinical history, you can plug the gaps from that 65% of people that have got bad antibody spectra with sotrovimab, Evershed, were the antibodies available at the beginning of the pandemic, but sipavibart is the one that's out there at the moment, made by AstraZeneca, and we're trying to get hold of this in the UK. We're chatting frequently to AstraZeneca about getting it, and it is available in Europe. It's not yet available in America. The other option we tried is a vaccine. This is a protein vaccine, not a messenger RNA vaccine. And it is made by Novavax Sanofi, and they had it out for a while. We tried it in a few patients, and I'll show you those results in a minute. But it's now no longer available. But it does mean that 35%, the hyperimmune and the unclassified group, need to go to the doctor now, because the hyperimmunity will give the doctor some idea about what to do. The unclassified might be because it's Lyme disease, it might be because it's Epstein-Barr virus. And the doctors with whom we're collaborating, people like Dr. Ben Sinclair, Dr. Finleys, and Dr. Benita Kane, they're coming up with tailored responses to those options. So there may be ways to manage these, even if I don't know yet as a scientist what's causing them. So this is what the Evershed did. Here's a patient here, reasonable antibodies, but a gap in their quality. We gave them Evershed. We gave much higher concentration of antibodies that trebled the antibody concentration, good quality antibodies. This patient was bed-bound in their bedroom, not awake for very long during the day, migraine every day. By the end of the first jab, their gut function had improved significantly. Their migraine frequency had dropped to 1 a month, and they started getting better. And we started measuring, here's the concentration rising over time. And after the third jab, they felt so much better. They ruined my graph and went to Greece on holiday. And so it looks like this intervention for this patient worked well. And the bit that's really interesting, this is very rarely seen in science. This is the antibody spectrum in their blood, but this is the antibody spectrum in their cerebrospinal fluid. This is the whiskey-like liquid around your brain that shows that the antibody was getting into your brain, that shows we were putting enough antibody in their blood to get to the point where things like the brain fog could be treated. So that's a really exciting result. We need to do that more often. We've done antibody interventions on 2 people, and there are lots of people interested in getting antibody interventions if we can get it into the UK or get to the patient to Europe. Novavax we've tried. So here's an antibody spectrum of a patient, long COVID patient, gaps in their spectrum for quality. After one jab of antibody, of vaccine rather, we repaired their spectrum. They started getting better. They had 3 jabs to make sure these levels got higher, and they're showing an onward trajectory in the improvement of their symptoms. So that's quite encouraging. This patient, however, is hyperimmune. This antibody level doesn't change very much. And she was going to hospital once a month or so with complications around her condition. Once the doctor saw it was autoimmune, potentially autoimmune, interventions around steroidal to reduce inflammation and other drugs to associate to remove the autoimmune response were considered, and her position is now being managed really carefully. Viv is very excited about the fact that, first of all, we showed that there was a problem, and before people just weren't believing her, but also after that, it got managed properly. So that's what the Attomarker test is showing, and these treatment options for people. And that triggers all the questions, Jonathan. So I think we should.
There's quite a lot to unpack there, Andrew. Thank you very much. And that actually answers quite a number of the questions that we've been asked. There's a couple of detailed questions here, which I think are very interesting. I should preface this with the fact that obviously Andrew is speaking as a scientist and coming from a scientific perspective. And if you're looking for clinical advice, please consult your doctor. And indeed, we will be having future webinars with doctors participating. So I hope you look forward to that. Here's one question. There was a recommendation I saw that a 60% weak antibody cohort should take Novavax 3 times over MABS. Has this changed? And has anyone gone with that weak antibody cohort, gone through the 3 times Novavax? I hope that makes sense.
So 3 times Novavax is the vaccine intervention here, and this patient, this is what their antibody spectrum looked like before Novavax, and this is what it looked after the first jab, and after 3 jabs, the antibody levels were much higher. So going back to why I need the antibodies to be everywhere in the body, and the concentration just needs to be high enough, and we got it up in this patient, I think, after the 3rd jab to more like 100 milligrams per liter. The quality is not bad from Novavax, around 40–50%. That means it'll hang on for long enough and the clearance should work. Now, Novavax I like because it's a protein vaccine. It's a very traditional vaccine. It's the same as you would have had in your arm for BCG or measles or some such in the past. We know how it works, and it's a reliable technology. It's not the messenger RNA vaccine, and it specifically makes good antibodies, and it should solve the problem. It should help complete your antibody spectrum, repair it, and then clear the virus. And I think from the 4 interventions we've made so far, those patients are showing better improvements.
Great, thank you, Andrew. So again, questions coming in quite thick and fast here. How far away are we from treatment being available on the NHS?
The NHS is going to require a data set that shows that the interventions work. They need to see from us that the clinicians are finding it useful, the doctors are finding it useful. And I think in the case of the antibodies, we need to collect a cohort of 50 to 100 patients to show that the outcomes look promising. As I said, recovery is an interesting question. We've had patients with really complex symptoms 5 years into a long COVID journey. You might argue it could be 5 years out of a long COVID journey. It could be a slow recovery. There's a risk that some damage and that recovery may be partial but stabilized. But we have seen patients that have had Lazarus recoveries within a week, gone from a wheelchair to back to better. The NHS will need to see a large group of people. They will probably want to then run a multi-center trial. At that point, it's quite expensive and it's going to take 2 or 3 years, I would imagine. If we stir up a fuss in the private clinics and get some good results, that may accelerate adoption maybe in other countries before the UK.
Thank you. We've got a lot of questions to get through here, so I'll continue on. Are other Attomarker tests being developed for co-infections seen in long COVID and other infection-associated chronic conditions?
This is right at the frontier of what we're doing at the moment. We think we have access to a new universal Lyme test. So Lyme disease is a tick borne disease. You get bitten by a tick and it puts a bacterium in your blood. Often people make bad antibodies, although the test is not good for that. And Lyme can become persistent. Now, why is it persistent? I think it's because the antibody quality is not good. And I have a feeling this might be long flu. It could be shingles over chickenpox. It could be Epstein-Barr virus and ME. This idea of characterizing the clearance is a new thing. And I think the application of Lyme and potentially EBV, we're exploring with the ME charity, Action for ME. I think we have an option here. And by later in the year, if we get the things funded, either in the research group or in the company, yes, we'll try and bring those out to the market.
A lot of questions, Andrew, about treatment availability. You've answered that in part at the start, but here's a more specific one. Will the latest Pfizer KP.2 vaccine help with building immunity like seen with the Novavax vaccine? Have mRNA vaccines shown promise for helping fill this antibody gap post-COVID and help with long COVID?
So the test here goes out to, as you can see, the variant JN1. We've also added KP3 to it, and that's the target for the vaccine. That means a message has been written specifically to make the spike protein that comes out of the JN1 or the KP3 variant. If the vaccine works well, as it has done in trials, you would make very good antibodies to this variant. And if that also finds a patch on the surface of spike protein, you could make antibodies to all the variants. We have tried giving messenger RNA vaccines to people. It does work in that we've seen antibody spectra being repaired. But amongst the long COVID community, there's a real anxiety that vaccine injury has caused their long COVID. And there's plenty of reporting in the literature about the messenger RNA being found around the body where it shouldn't be.
Thank you. Are there any trials available moving forward for COVID vaccines such as Novavax?
When we're chatting to Novavax, I exchanged emails with them today at Sanofi. When the new version is available, and FDA announced yesterday that they're requesting some more data for the latest version. When that's available in the UK, you'll be able to buy it. I took Novavax twice last summer when the old version was available. So my antibody spectrum does look like this. I took it because undergraduates tend to return to university with all sorts of flu within the first 4 weeks, and I didn't want to get COVID. When it's available, you should be able to buy it privately. Again, if your antibody spectrum suggests that's a good intervention and the doctor thinks it's a good thing to do, why could you not try that? And again, I would recommend 3 shots of the virus, probably a month of the vaccine, probably a month apart to get the antibody levels up, and track the symptoms and track what happens. Given what I was saying about recovery, you need to know that it's worked or not, and the doctor can help interpret that.
Yes. So quite a complex picture right now in terms of treatment availability and perhaps some almost political advocacy as well as medical advocacy required, given that some of these treatments are available in Europe and not in the UK. I noticed that Novavax has just applied to the FDA for further approval. Do you have any comment on that, Andrew?
I think adoption, and Novavax is more likely to be adopted because of the price. It's around £50 a shot, whereas Sipavibart is going to be more like £2,500 a shot. So if we could show Novavax worked, the patient wouldn't have to leave their home. The risks for getting the vaccine would be rather low. You could send around somebody to vaccinate, and slowly people would get better at home. And I think it's very attractive to try that. Let us try it with some patients quickly, based on classifying them correctly with their antibody spectrum. Would I give Novavax to a hyperimmune patient? I think the doctor would want to make that decision, but I can't see as the crazy scientist why that would help. But the doctor may decide otherwise. So let's get some interventions. Let's publish the data quickly. And we are looking to engage with the UK HSA and government as quickly as possible to share the data.
Yes, some really detailed questions here, and it shows how well informed the long COVID community is about the situation and, of course, their own condition. At this point, I think we've got another poll question to show. If that could come up, that would be very helpful.
Okay. Ah, would your doctor be interested in learning more about the Attomarker test?
Thank you very much. Right, let's go into some of these more detailed questions. And this might be like your viva voce at Cambridge, I believe, you for your first degree, Andrew.
Yeah.
I have to forgive you that. Updated vaccines target more recent and prominent COVID strains. If we still have poor immunity show in our diagnoses for earlier strains, such as BA1 or XXB 1.5, how can we now build antibodies for these strains without the old vaccines? This comes back to the hinge, targeting the hinge as well, doesn't it, as opposed to the spike?
Yes. So what we found with the interventions that we've made is that the Novavax targets for the later variants, targets a gap of the spike protein that's there for the later variants. But if it's there for the later variants, it may also be there for the earlier variants. It's possible that the gap is not conserved, and that's important, the patch where the antibody attack's not conserved. But it's likely that you'll make good quality antibodies across the spectrum. Of course, we can tell you that if Novavax were to make good quality antibodies to the earlier variants or the ones that are causing your persistent virus, we'd measure that, and then you'd measure the change. The hinge bit is this bit. This is the spike protein. These are all the blobs that have happened in mutation. What's not changing is this bit. And that's really interesting as a chemist, right? So there are parts of the protein that don't change, and it's because that's a hinge. And what it does is it's floating around the surface of the protein, like of the virus, like this. But when it binds to the receptor, it makes this big change. If you break the hinge, it's not going to work. So I've been advocating for some time we need a hinge vaccine that targets this region, or an antibody that targets this region. And until the virus works out how to build a new hinge, that will work for all variants. That's coming up as a suggestion in one of the papers shortly.
And when are the papers due out, Andrew?
3 of them are on the pre-pub server now, and the long COVID, and now going to the journals for peer review. The long COVID paper, classification papers now with the authors for final review, that will go to the journal next week, I hope. So depending on how excited the journals are to publish, the data should be available fairly shortly.
Yes, we'll share those with the subscriber group so they can share them with their clinician or consultant. I believe that would be helpful.
Absolutely.
Very interesting question here. Someone who had an Attomarker test some time ago, which revealed an abnormally high level of antibodies to the first 3 COVID variants. They got monoclonal antibody treatment with Eversheld and have seen some benefits during the last 7 months. Is there any benefit to repeating the Attomarker test now to see if the levels of antibodies have improved, or will the test still see antibodies from the Eversheld? Is there any other way of monitoring my progress?
Lots of questions.
Yes.
So if you've had Eversheld, Eversheld works really well against these early 3 variants that we have in the picture here, and you can see it responding in this patient. And there's a big drop. It doesn't work after that at all. It doesn't work after Delta at all. So it's targeted at a part of the spike protein that's no longer there. So Eversheld needs to get to a high enough concentration in your blood to get round to all the corners of your body where the virus may persist. And that's why you're seeing improved symptoms. But I think that improvement will slow as the concentration falls off. The antibody has a half-life, so the concentration of antibody in your blood for Eversheld halves every 40 days, which means the effect will begin to slow down and will last for a period of time. Which is why with this patient here, we gave the Eversheld 3 times 40 days apart, so that we get the really high antibody concentrations, especially to get it into the brain here. So that's why the spectrum protocol we're proposing to the clinicians to turn my crazy science into medicine forces the antibody concentration to rise to get around the body. I think the patient who has had Eversheld would benefit from Sapavibart. I don't think there's a problem with the conflict of those 2 drugs. The fact they've had a response means the clearance is working. And if they've already had our test, I'd really like to know who they are, because we might put them in the case studies paper that's coming up shortly. So I would suggest that's good evidence to keep going, and Sapavibart will be the next available option, I think.
Yes, I understand the test is currently CA marked with a view to CE marking and FDA approval in due course. Is that correct?
Yeah, so in the UK, the regulation is a UK CA mark. It replaced the CE mark when we left the EU. We are applying for the CE mark as well. It's already performed on a device that got a CE mark, so we were confident about its reproducibility. And that will then get, I think the test will get the CE mark later this year. We are submitting to FDA to show that the test works, and that's now being prepared. And our pre-submission will go into the FDA, I think, even this week. So yes, we're looking to share the data with everyone that we think is important.
And related to that, from some of our subscribers living outside the UK, can people from the EU or the USA or India do this test, or do they have to be based in the UK?
The only question about coming from far afield is the sample's got to be in reasonable state when it gets here. So we have shipped samples back from the States. We have a partner that offers what's called cold chain, and it can ship up to 40 samples back at once. And that would cost about £60 per sample, which isn't too bad. But obviously, if it's just your sample, it's going to cost about £1,200. And that should also work from India. That should also work from the rest of the world. So if you can get 40 friends together or 20 friends together, the cost of shipping isn't going to be too bad. In Europe, DHL do a reasonable job for us and some other distributors as well. So we can get the sample back in good shape in a day or so, and it still works. So as of the moment, getting it back to Attomarker is not a problem. If we get really exciting information, we'll be looking for partners in America and around the world, because the test is not difficult to run now that it's got a UK CA mark, all the documentation's in place. So we should be able to do it in lots of different places when it becomes the standard in treatment.
Thank you. We've got 3 or 4 minutes left, so I'll try and get through as many questions as we can in the time. Here's a question around co-infections. So can people with co-infections or people with ME benefit from this test?
Yes, so I think in general, long COVID patients appear to be complicated, which is why the doctors use our test to differentiate between our infection and other infections. So if somebody had a good antibody spectrum, but had chronic fatigue, for example, they may have chronic Lyme, they may have ME, but at least you know their antibody spectrum wasn't needing to be repaired. But we do know some people in the clinics that have got very bad antibody spectra, but have also tested positive for Lyme disease. And so they are on a series of a course of antibiotics, which is a long journey to remove the Lyme. And then after that, we would repair the antibody spectrum. But that's why the doctor needs to get involved. The more complex the patient becomes, resurgent EBV, Epstein-Barr virus, resurgent herpes virus leading to genital warts that's been seen on patients getting better. Once your immune system seemingly is compromised with long COVID, all other viruses that are persisting can then come out to play. And I think it then becomes a complex, multimodal symptom problem. And the doctors need to use our test to at least eliminate part of that.
Thank you so much, Andrew. So some information about up here on screen about the test and how to get the test. And also we have a further webinar, which I think some of the questions that unfortunately we've not been able to answer during the course of this 30-minute session will be answered, hopefully, by Dr. Finlay, Dr. Ben Sinclair of Dr. Finlay's and Professor Andrew Shaw. Can everyone see that? Yes. So there's the information on the forthcoming webinar. And then if you want to buy the test, this information is also on the website, and you can see it there. So a survey will be sent to you, and I'd be most grateful, we would be most grateful if you would complete it, how you found the webinar, how informative it was, some suggestions about the future. And we do hope that you keep in touch with us because we believe very strongly in well, the benefit of the test, but also a focus on patient outcome. Often a concern, having direct experience with long COVID myself, that the patient is not the most closely regarded element in the process, and we Attomarker, we want to change that. Andrew, is there anything that you'd like to say in conclusion?
It's very important for the medic to help turn my crazy science into medicine. It's a difficult problem, but scientifically, some doctors have been telling us that it's the most logical explanation for long COVID that they have seen. The test aligns very strongly with what's in the literature. So I think it's important, and I want to get people to get an intervention, get tested, get the intervention, and we will publish it quickly and raise the profile internationally.
Well, thank you so much. And thank you to Steve and Suzanne for helping in the background. It's been a very interesting session, and we look forward to joining you again. Thank you. Good evening.